Hello, everybody. My name is Mitch Bandy. I'm from Hendrix College, and today we'll be talking about YtfE's reduction of reactive nitrogen species. YtfE is a protein found in E. coli that also has homologs in many other bacteria. The presumed function of YtfE is that it is utilized within the cell to reduce reactive nitrogen species, or RNS, and repair iron-sulfur clusters that are damaged by RNS. The active site specifically is thought to be responsible for reducing the NO and donating one of its iron atoms to damaged iron-sulfur complexes. The structure of YtfE is shown on the left. However, for what we're discussing today, the active site is of more interest to us. The active site of YtfE on the right is situated within the heme-erythrocyte domain, or the blue domain, and consists of a mixed valence diiron center of iron 2 and iron 3 respectively. It also consists of a mu oxide bridge, shown in red, and two carboxylate bridges constructed of glutamate 133 and glutamate 208. Each iron is a 6-coordinate iron atom with only 5 sites bound and the unbound coordinate being trans to the mu oxide bridge. The irons are coordinated to the protein through different histidines, with iron 2 being coordinated by histidine 84 and histidine 204, and iron 3 being coordinated by histidine 129 and histidine 160. The importance of this research specifically is to be able to better determine how YtfE prevents nitrosative damage and repairs damaged iron-sulfur clusters. If we can determine the mechanism for how this proceeds, we can better understand how to combat bacterial diseases as the human immune system produces NO as a defense mechanism. Studies have shown that YtfE can reduce NO to N2O, suggesting that the enzyme plays an important role in protecting E. coli from the damage due to nitrosative stress. However, the exact mechanism is unknown, and thus our hypothesis was that NO reduction is the primary function of YtfE. In order to test the hypothesis, we first determined the kinetic rates of the reaction, as that is the first step in determining the physical mechanism. The reaction scheme for NO reduction, shown here on the left, has two nitric oxides being reduced with two hydrogen atoms to produce two nitrous oxides and a water molecule. This is made possible through the oxidation of the YtfE active site. In the figure on the right is the UV-vis data from the iron site. The reduced YtfE has a feature at 352 nanometers where the arrow is pointing, while oxidized YtfE does not have this feature. The EPR, shown in the inset here, proves this. However, that will not be part of this discussion. In other experiments, we have shown, using EPR data, that the loss of the reduced YtfE absorption signal is correlated to the loss of NO concentration in the cell. We will follow the decay of the signal at 352 nanometers to determine the kinetic rates of NO decay by YtfE. Following the decay of the reduced YtfE signal, we obtain a kinetics graph that conveniently follows a Michaelis-Menten plot. Here are the curves for 3 different enzyme concentrations with the fit lines and data points. This is one representative data set, but the experiment has been repeated multiple times, and the results of the fits, Kcat and Km, are found in the table. Km is representative of the substrate binding efficiency, so the high Km values show that we have a complex that readily binds. However, the low Kcat values are representative of a slow reaction compared to the production of NO by the immune system, therefore telling us that this reaction, while YtfE and NO bind efficiently, does not have a high rate and proceeds slowly. Based on the average Km derived from the experiments, YtfE can bind any NO it encounters at these concentrations. While this is the truth, Kcat, the turnover number, shows us that YtfE is not at all efficient in reducing the NO to N2O, with the catalytic efficiency being approximately 0.05 inverse seconds. When we compare this to nitric oxide reductase, which has a Kcat of about 10 times higher, a Km of approximately 10 times lower, and a catalytic efficiency of approximately 8, we can see that YtfE is significantly slower in its reduction of NO. Based on these results, our hypothesis that NO reduction is the primary mechanism of YtfE seemingly is false, and that NO reduction is in fact a secondary mechanism for YtfE. Future directions of this research include determining the physical mechanism for how YtfE reduces NO to N2O, whether iron-sulfur cluster repair is YtfE's primary function, and the physical mechanism for iron-sulfur cluster repair by YtfE. I would like to thank Hendrix College, the principal investigator, Dr. Bill Gunderson, my co-researchers, Kosti Shaw, Michael Miller, and Cole Bolin, along with Dr. Brad Pierce of the University of Alabama, Tuscaloosa, and I'd also like to thank Arkansas and Bray for funding the project through a research and development grant. Thank you very much.